Well, welcome everyone to the first RED Talk, the Rensselaer's Research, Education, and Discovery Talks. You're going to hear just a little bit of some of the work that's being done in research and how that research also links to what we educate our students in. You'll hear from anything from climate change and geological era to what's happening in rebooting medicine, to opportunities and how you deal with lots of data, but then using it to make things better and to provide better opportunities to get products to you, to learning about how art and science come together, and then finally how you perceive things. And if some of you were in the CISL this morning down at MPAC, you'll hear a little bit more about some of those things. But the ability to bring cognitive and immersive systems together, as well as to see what in the world is going on with AI. So we have 5 presentations today. What we'll do is they'll be TED-like, but obviously we call it RED. And that in addition, I know there may be a lot of questions, but we'll go straight through, and at the end, hopefully there'll be a little bit of time, and we'll be able to have questions asked to any of the speakers that you would like. So with that, again, thanks for coming, and let's get started. The first presentations will be given actually by 2 faculty in the Earth and Environmental Sciences Department. Karyn Rogers, who's a professor, of course, in Earth and Environmental Sciences, and Peter Fox. Peter is a Constellation professor, the Tetherless World Constellation, and they're going to talk about what about our planet, what's our planet telling us. Well, actually, you can see the title. The planet is telling us something. Will we listen? Obviously, I didn't, but they will, and we'll start right now.
Good afternoon and welcome to the RED Talks. My name is Karyn Rogers.
And I'm Peter Fox, and today we're here to tell you about how we listen to the planet. And at some point, we'll get a clicker so that we can advance our slides. It's a professor thing, you know, it happens from time to time.
So it turns out this idea of listening to the planet isn't just the title of our talk, it's actually my day job.
So Peter Fox, I'm an applied mathematician. I do data science, I do environmental informatics, and
I'm a geochemist, a geomicrobiologist, and in some cases, best of all, an astrobiologist, which means I get to spend my days trying to understand how the planet and life have evolved over 4.5 billion years. And so part of that is understanding the origins of life. And I was recently, I had the opportunity to go to an Origins of Life conference in San Diego in July. And my job was to explain to my colleagues exactly what it was the early Earth looked like almost 4.5 billion years ago. So I was taking them through this litany of sort of characteristics. So one of them is that the atmosphere on early Earth was probably 100 times more dense than it is today. That atmosphere had no oxygen, and it was primarily filled with CO2. There were oceans, but those oceans were somewhere around 70 degrees C, which is nearly 160 degrees Fahrenheit. And I was going through this long list, and my colleague Niles stopped me. He said, Karyn, wait, stop. I don't understand. He said, when I came up studying the origins of life, that's not what I understood to be the early Earth. You see, Niles came up under the paradigm of Miller and Urey. So Stanley Miller and Harold Urey in the early 50s did this really groundbreaking experiment, and I think some of you probably recognize their apparatus because it's littered through all of the biology textbooks that we have today. And what they did is they thought the early atmosphere had methane and ammonia in it, so they put it in this apparatus, they induced a spark discharge, and they made amino acids, which are some of the fundamental building blocks of life. And this is what confused my colleague Niles, and he stopped me and he said, Karyn, this is what I learned, and that's very different from what you just told me. So what happened? I said, well, Niles, we learned things. In 60 years since this experiment was done, chemists and geologists and a number of physical scientists have figured out how to better listen to the Earth. And so in the next 20 minutes, what we're going to do, we're going to take you through 4.5 billion years of Earth's history, and we're going to try and sort of show you what we've learned about how the Earth and life have changed together through time.
I know you don't have actual seatbelts on your seats today, but you might find that if we're going to take you through 4.5 billion years and 27 orders of magnitude in spatial scales, you should buckle your seatbelt. So Earth is positioned in the spiral arm of the Milky Way galaxy, 10 to the 21 meters of order. Here's a rendition. You're going to see this artistic rendition because we didn't have pictures of the Earth back then. 4.4 billion years ago. Karyn's going to tell you a good story about how we can listen to the early Earth. And within this Earth system, because it's a system-level problem that we're really trying to attack, there might have been quite likely hydrothermal vent systems, which we know exist today, which have high temperatures and pressures and interesting chemistry. And then within these communities are microbes, microorganisms. And this is an example, red and green, are two communities that have found a way to coexist under extreme chemistry and physical conditions. And so this is 27 orders of magnitude. Now, as we go back in time, 4.5 billion years ago, there's the early Earth. And today, on the top of the axis there is this microbial community, 27 orders of magnitude in scale and 4.5 billion years of time. And we might think today that we're in this wash of data, and we are in this wash of data, but it's really about how we extract the data across this entire timeline of the age of the planet.
So we're going to start with the very beginning of Earth's history. So this is an artist's rendition of what the Earth looked like probably 4.4, 4.5 billion years ago, recently after it accreted. And what we'd really like to know, if we want to understand how it is that life might have emerged on this type of system, is we want to know some very basic things. Was there water? Was there land? What was the atmosphere like? In order to answer these questions, we need to be able to learn things from the very few pieces of early Earth we have left. Now, I have the privilege, when I have those sorts of questions, of simply walking down the hall. And I get to ask my colleague, Bruce Watson, who is a professor in Earth and Environmental Sciences. He's also an Institute Professor of Science and a member of the National Academy of Sciences. And Bruce has essentially made his career by looking at the very few pieces of Earth we have left and asking those pieces questions and getting them to give him answers. These are pictures of zircon minerals. They are the oldest material on the planet. They're found in Western Australia, and they are as old as 4.4 billion years. That's just a little over 100 million years after the moon formed and 170 million years after the Earth formed. And so through painstaking work over decades of both experimental and field work, what Bruce has been able to do is figure out what these zircons are telling us. And they're telling us some really interesting things. Within 150 million years of this planet essentially being a ball of magma, there was water on the surface. It had cooled enough for there to be water. There was an atmosphere that was made almost entirely of CO2 and nitrogen that was much more dense than it is today. And there were continents, or at least land masses. And those land masses looked a little bit like the land masses we have before us today.
So chemistry was happening. In the early Earth, the chemistry was active. The Earth has changed over time. Its physical structure as a system has changed. The continents have come and gone. Oceans have opened and closed. And the minerals, zircon is where we start from the very early part of Earth history. But we have 5,700 minerals. The first 500 or 600 were made by chemistry. The rest were made by biology. So by going in and looking into this mineral history, the concept of mineral evolution over time is where we start to ask questions from the Earth. At the same time, the atmosphere has changed. The two major increases in oxygen in the atmosphere were fundamental to the origins of life and biological organisms. And you'll see it's a fuzzy line. So we've got minerals, we've got composition of the atmosphere. What can we learn from the Earth by asking it questions? Well, it turns out that one of the new techniques is that minerals have a social life.
Sometimes their social life is better than ours.
Yeah, it's actually much better than ours. So what you see in the top here is, over time, a network diagram of the co-occurrence, which minerals like to hang out with other minerals. And how that evolves over time tells us a lot about where they were formed, how they were formed, and who they hung out with, and what are their similar chemical properties. So network analysis, we learn things from Facebook even. As the Earth has changed, and as the minerals have evolved over time, so here is a skyline diagram which represents the sum total of all of the data around minerals containing fundamental metal elements. And so we go from cobalt all the way through to iron and copper. This is, again, 4.5 billion years of history. The images of the Earth here show you the opening and closing of the continents, and you'll see that minerals don't exist forever. They can go extinct. And they come and go, and they line up with the opening and closing of the continents. And we've learned tremendous things about this. But metals don't just show up in minerals. They also show up
in proteins. And so you might think, if through all of this period of time, the continents are changing, and the atmosphere is changing, and even the chemistry of specific trace metals is changing, I wonder if that has any impact on the life that is also evolving with the planet at the same time. And it turns out that it has. So what you're looking at here is the social network of proteins. Now, proteins are the thing that makes your cells and my cells and every other cell on the planet do their things. They are what makes cells actually function. And as the availability and chemistry of metals on the planet changes through time, it turns out we see that chemistry reflected in the chemistry of proteins. And so the early proteins were primarily centered on iron, and the later proteins are centered on copper. And so we can see this chemical evolution that is planet-wide, actually in these very, very tiny molecules that also allow life to exist and continue. And so we see these changes on both very broad scales, broad continental and atmospheric scales, and we see these changes on very small scales at the level of tiny protein molecules. And this is absolutely fascinating. But all of these things are global. Everything we've talked to you about so far is at the planetary scale, the giant global scale over 4.5 billion years. Is there anything perhaps that we might want to know on a slightly smaller time scale or a slightly smaller spatial scale? Well, of course there are. There are all kinds of things that happened locally in very short periods of time in Earth history. One of those things was recently sort of discovered by my colleague Morgan Schaller, also down the hall from me in the Science Center. And so what Morgan does is he tries to understand how the Earth has changed due to very short-term events. What he's actually recognized by looking at 4 different samples off the east coast of North America is he has discovered a comet impact that occurred 56 million years ago. This is just 10 million years after the dinosaurs go extinct, and he has found in these 4 locations evidence of a comet impact. Some of that evidence are these sort of impact spherules that come out of the ejecta from this comet hitting the Earth. And some of that evidence is sort of these charcoal samples that are evidence of global wildfires. Now, as part of this comet impact, there are all sorts of things we can learn by looking on these very short spatial scales. And one of the things that immediately followed this comet impact was a doubling of CO2 in the atmosphere, an increase in global temperatures of nearly 5 degrees, and probably ocean acidification and a relatively small global extinction. If any of these things sound familiar to you, it's because they're the exact same numbers we're looking at today for global change. a 5-degree change in global temperatures, a doubling of atmospheric CO2 on relatively short timescales. If we want to understand how the planet might react to that today, one of our best bets is to look into the past and try and understand how the entire Earth system reacted then.
That's impressive, I think, don't you? So at the heart of this, we have a variety of techniques when we listen to the planet, going back from the early zircons, looking at the social life of minerals and the spherules, and bringing up to the current day, and again, going back to this characteristic that the amount of data over these time range and these spatial scales is increasing in an exponential sense. But we don't quite have all the data that we need. At the top end of the spectrum, at the microorganismal scale, we have gene sequencing. We can generate so much data so quickly, but if we're going to generate that data not from a human, but from the planet, to get a representation of the whole planet, we need to sample the whole planet. Well, we can't quite do that. We have limitations on how much we can do.
If you think about what we are asking here, is we are trying to understand environments at a global scale and life at a global scale. But that life experiences those environments at the order of magnitude of 1 micron. So we're trying to understand global scale sizes at the size of a single cell. And if we want to understand how geology and biology interact, we need to be able to figure out how it is these single-celled organisms react when there are changes, both at the local scale and at the global scale. And despite all of the genome sequencing data we have from microbes and humans and everywhere in between, we are vastly undersampling the life on the planet. And so as part of our recent collaborations here, we're asking very new questions. And one of the questions we're asking is, how is it that biology reacts to these environmental changes? And we want to know not just about individual organisms, but communities of organisms like this community that's shown here. And we also want to understand that at every possible environmental condition you can imagine that could ever exist on this planet. That's an impossible sampling question. But if we move this into engineering scales, we can fix this problem. We can actually ask the question, how is it that groups of microbes respond, not just in who they are, but how they function, and how they change environmental conditions when those environmental conditions change? We can figure this out by bringing these organisms into the lab, scaling this up with our engineering colleagues, and figuring out how it is the functional microbiome works. So this is a brand new project that goes between myself and Peter and our colleagues in engineering. One of them is Cynthia Collins. You'll hear from her next. And we're also working with the IDEA group, and so you'll be hearing from IDEA in just a few minutes. And what we're trying to do is really push the boundaries of what we can ask life about how it responds to changes on the planet. One of the hardest things to do in this project is to make sure we can replicate every sort of environment on the planet in the laboratory. The hardest thing to do, it's not pH, it's not temperature, it's not salinity, it's not lack of oxygen. The hardest thing to replicate in the laboratory is high pressure. Now you might think, why do I care? I live here on the surface of the Earth. But 93% of all the single-cell carbon on the planet lives at high pressure. And the early Earth, 4.4 billion years ago, all of it was at high pressure. So what I do in my lab is I bring microorganisms into my lab and I ask them, how is it that you deal with changing conditions? And particularly, how is it that you deal with high pressure conditions? And so when I do this using traditional methods, That's what happens to the microbes. They die. And I'm not a coroner, so I can't get the dead to talk to me. This is a very disturbing thing. And so what did I do? I invented a way not to kill them. And so through some collaborations, international collaborations I have, we have developed this new device. It's called the pressurized underwater sample handler. It allows me to grow these microbes in my lab at elevated pressure conditions. And when I do that, they don't die. They grow. And when they grow, I get to interrogate them. And that's sort of the best part of my job. This is absolutely phenomenal. We are about to take this device to the bottom of the ocean to 5,000 meters, not just to ask questions, but to get new microbes, so I can ask new microbes questions. So what we've developed so far, the push, it prevents death. I promise you, in the very next generation of the push, I'm going to work on preventing death and taxes, but I started with death. So I hope what we've shown you in the last 20 minutes is that we've come a long way. This is the image we had of the early Earth in the 1950s when Stanley Miller and Harold Urey did their groundbreaking experiments. And since then, we have made fundamental changes, and we have shifted the paradigm of what we understand about the Earth, how it evolves, how life got started with it, and how life evolved since then. And what we're doing here at RPI right now is that we are pushing the boundaries to get to the next step. We are being innovative and cross-disciplinary so we can ask the Earth the next set of questions. And I, for one, can't wait to hear what the planet has to say.
And I'm excited too. Thank you for listening.
Thank you.
Thanks, Karyn and Peter. So anyway, we've heard a little bit about the microbiome. We're now going to hear a little bit more about that, in particular when we talk about rebooting medicine, keeping in mind that there are many more microorganisms in our body than there are cells in our bodies. And then the impact of going from microbial systems then to actually making molecules that are helpful to us. Our next talks are given by Cynthia Collins, who's a professor of chemical and biological engineering, and Robert Linhardt, who's a Constellation professor in biocatalysis and metabolic engineering and a professor in the Department of Chemistry and Chemical Biology. So.
So I'm Cynthia Collins. I'm a professor of chemical and biological engineering here at RPI, also in the Department of Biological Sciences. I started off my career actually as a chemist, and somewhere along the lines I decided I really like to apply problems. I even did some microbiology along the way. And so I was the interface of engineering and biology and chemistry. And so I'm going to tell you a little bit about what's going on at RPI in that realm. And so when we think about problems in this round, we think about engineering biological systems. This is pretty much what we think about, right? Trying to take tools like a hammer or a wrench and trying to engineer biological systems, where essentially every time there's a problem, we come up with a new tool, a new solution, right? And from an engineering perspective, right, this isn't very satisfactory a lot of times, right? We really want rules and tools and design schematics that we can essentially go back to develop any new system we want and solve any global challenge, right? And there's amazing global challenges in the realm of biological systems, right? And so some of them are illustrated here, right? And so we can think about changes in agriculture. So can we design systems that are less reliant on energy and water? Can we design systems for new healthcare systems, right? Can we develop new drugs? And can we change our reliance on petrochemicals, right? Make things more biological solutions, everything from oil and fuels to materials, right? And so to do this, we can use what we call synthetic biology. And this is a new approach developed maybe 10 years ago where we try to treat biological systems with essentially engineering concepts. So can we plug in? And so these approaches have been a little bit about, right, addressing challenges, kind of follow that included all the amazing groups at RPI actually think about microbial communities every day, and sometimes we think about engineering them. And so entire career on one microbe or one protein, right? I think that the way to go is to keep thinking about microbial communities as we think about engineering processes. So why do we want to do this? So we can divide labor between multiple organisms, take advantage of their strengths and combine them. I need this. I'm not going to give away everything. All right, see if we can get this to work. Can everyone hear me better now? Okay, all right. Okay, so divide labor, microbial communities, right? In this building, we're not all jacks of all trades, right? We all have specialties, amazing specialties as RPI alums, right? And so just like that, we want to divide processes amongst different microbes in order to have them carry out synergistic processes, whether or not it's making new materials, right? Making new fuels, making new pharmaceuticals, or materials. All right, so I'm going to tell you a couple of vignettes along these lines. The first one is from my research group and talking about how we essentially develop processes using mixtures of microbial communities. And so we think about a few things in order to get these processes to work. So one of them is, how do we engineer organisms to grow together? So in this realm, we've been essentially designing organisms where one organism requires another for survival. And so we can use these kind of strategies, what we call mutualism, in order to make organisms require each other. Something that's been really fascinating to us is that if we essentially pair these organisms where they have to essentially sustain each other, provide a chemical that the other requires for growth, they actually are doing this, and we can see long-term paired growth, co-culture growth of these organisms much better than we expected. And so that actually tells us a little something about how organisms might exist in nature and how they're actually kind of tuned and primed to actually collaborate in this kind of way, which is really exciting. The other thing we think about is, how do we make cells coordinate their behavior, right? You wouldn't have a large expanded process without having communication links between each of those nodes. And so we can take advantage of communication systems that bacteria use, that they generally use to sense whether or not they're alone or in large population densities, and change their behaviors accordingly. We can use those kinds of systems to coordinate behavior amongst organisms. And we've been pioneers in engineering different ways to make cells communicate with each other. And the last aspect, of course, is, what do we want them to do, right? The traditional, right, pathway and reaction engineering that you might use approaches like synthetic biology, metabolic engineering for. And that's, how do you divide a process amongst different microbes? How do you take advantage of their strengths? And so an example of that is work that my group is doing, where essentially we're trying to go from plant waste, so cellulosic biomass, to essentially some sort of pharmaceutical or biofuel. And so we can divide this process amongst two different types of microorganisms. One is a set of bacillus organisms, and what they do is they secrete cellulases. And these, right, proteins can degrade that cellulosic biomass into sugars. And so we've engineered systems where we secrete those proteins, and those proteins actually work synergistically to break down that plant waste. The really cool thing is that in industrial processes, the production and purification of these enzymes is one of the most intensive parts of these processes. So now we're doing it in situ, right, in the reactor, and we can save significant more portions of the funds as we do this kind of process. Then the next step is to take those sugars and make whatever we want. And so in the current kind of setup we're looking at, we're trying to make different types of pharmaceuticals. In this case, you can make violacin. We're also making other compounds that are essentially antibiotic, antifungal, and anti-tumor compounds. And so we can use this type of process and modular approach to essentially ramp up and make different types of compounds from essentially waste products. So another aspect where microbial community is actually really important is in what we call the built environment, right? All of us are sitting right inside today. We probably all spend a little too much time indoors. Maybe we should roll around in the dirt outside or go for hikes at least a little more often, probably. But we're interacting with microbes every day, and more of those microbes are right from other people, right, might be healthy or non-healthy. And so there's lots of opportunities within this realm to think about how we might improve human health. And so I've had the pleasure of working with my colleagues at the Center for Architecture, Science, and Ecology at RPI, and they developed this amazing plant wall. And so this plant wall integrates with a traditional, right, air quality, HVAC type of system, but there are plants and microbes in the system that play a key role in improving air quality. And one of the cool things that we've been working on is essentially checking what microbes are living in on these plants. And we've been able to do some really cool high-scale data and analytics, and one of the fun things that we found is that organisms that are capable of degrading volatile organic compounds, which are right not so great compounds that we don't want floating in the air, are actually living in these plant associated microbial communities. And so these are things that we can actually ramp up and increase as we move forward with this type of approach. The last thing that you guys may have heard of, right, is the human microbiome. It seems to be everywhere these days, right, there's a new correlation between some sort of, right, health effect, from obesity to Parkinson's, et cetera. And so there's some great new work going on at RPI. John Dordick, who you've heard about a little bit today, he and I are working on essentially developing some new antimicrobial strategies that will target microbes within a community context, as are my colleagues Matthias Kofis and Blanca Berquera. And one of the great stories that's come out of RPI recently is from my colleague Jürgen Hahn. And in his group, they've been looking at essentially how metabolites can be used. So you have all of these metabolic profiles from different children, many with autism. And they found that you can actually correlate essentially metabolic profiles to autism. And so essentially this kind of approach allows us to develop new strategies, right, towards essentially identifying whether or not a child has autism way earlier than the traditional methods of diagnosis. And so, right, and of course many of these metabolites are due to the microbes, right, that are existing, right, in the patient's gut. And so I think I've told you a lot about synthetic biology, kind of got around to medicine eventually. And so now what I'm going to do is I'm going to hand the talk off, hopefully your microphone works, to my colleague Bob, and he's going to tell you about some amazing work within the realm of medicine and essentially addressing a key challenge using synthetic biology.
Thank you, Cynthia. And my mic does work. That's good news. Today I'd like to talk about a synthetic biology project, and I'm grateful to Cynthia for introducing both synthetic biology, metabolic engineering, and also microbial communities, because we're going to use some of those approaches as well. But we're going to focus on a problem. And the problem began with a crisis in 2007, 2008. Some of you may have heard about this. It was a contamination crisis in which heparin, which is an anticoagulant, blood thinner, was contaminated worldwide. And this contamination led to the death of about 200 Americans. And these are patients who are relatively ill or were in the hospital getting heparin, and they died from this tainted drug. So this crisis, like any crisis, motivates solutions. So I think Winston Churchill, this may be apocryphal, said that all good crises should be exploited to find solutions. And this crisis actually was solved in about a 6-month period of time. And that's pretty fast because this is a very complex drug mixture. It's a natural product that comes from an animal source. And this natural product basically was adulterated. And so it was an adulteration that took place in China, which is a highly unregulated country, or was at the time in this area. And it was adulterated with a relatively inexpensive substance called OSCS. And this substance was added deliberately to make money. So this cost almost nothing, and it could be added to the drug without ability of modern technology to detect it, and it was done solely for a profit motive. So it probably made the people who did this crime, probably made them tens of millions of dollars, but it certainly damaged the drug chain, not irreparably. So what we did in this 6-month period was we discovered what the adulterant was, and we discovered ways of assessing the drug to make sure it was clean, and ways of securing the drug market. So going forward, there would be less of this problem. So I have to backtrack a little bit and tell you what heparin is and where it comes from, because you have to know that and what it's used for, because you have to know that to understand why we're using this approach. So it actually comes from pigs, and it comes from the intestines of pigs. So the pigs are raised for food, not for their intestines, and the intestines are a waste product. And so 1.2 billion hogs are slaughtered every year worldwide. This is an amazing number to think about, are slaughtered every year worldwide to provide food to the world. And of these, 60% of these animals come from China. So it's a market that we can't ignore because this is the source of most of our heparin in the United States. And so this 60% market share actually goes into the preparation of a blood thinner, the anticoagulant heparin. And this is essential for the practice of modern medicine. Without it, there would be no extracorporeal therapy like artificial kidneys or kidney dialysis. There would be no open heart surgery. There wouldn't even be hip transplants or part replacements or major surgical procedures all require this life-essential drug. So it's essential for modern medicine. So we can't afford ignoring this problem. And here's the real crux of the issue. It's a mismatch between the food chain and the drug chain. So on one side of this slide, you see this woman with buckets of intestines coming from hogs at a slaughterhouse. And what she's doing is first they remove the fecal matter from the intestine, and then they scrape the lining. And that lining contains the component that goes into the manufacture of heparin. And that white slime that you're looking at in this picture is transferred to a pharmaceutical manufacturing plant that then produces a pure drug for intravenous injection into patients. And so you can see immediately that these two environments are regulated very differently. And this mismatch really makes it prone to have complications like adulterations, contaminations, viral impurities, all sorts of possibilities. So as I said, we learned a lot of lessons from this crisis, and that's what a crisis should lead us to, is lessons and solutions. And I'm going to focus on one of these, and that's really simple. And certainly being in a biotechnology center like this, like CBIS, we asked the question, could we source heparin from non-animals, from non-animal sources? Could we make it in a biotechnological process? And this is quite a challenge because it's a very complex polysaccharide-based drug, and it's a big challenge to do this. But we thought that this was possible. And the second question is, if it's possible, what's the scale that you have to think on? So this is a major drug, $7 billion a year in sales, 100 metric tons annually. So that's a lot of this material that you have to make in a biotechnological process. And we're fortunate that the market is subdivided in different forms of heparin. And so some heparins have a larger market share and some have a very tiny sliver. So this allows us the opportunity to segment the market and go after short-term solutions, intermediate-term solutions, and long-term solutions. And the long-term solution would be to eliminate the animal source drug from all applications. The short-term solution might be to just target a few applications in the immediate term to try to resolve this issue. So we learned how to make heparin biotechnologically from the cell that makes it in the pig. And so molecular biology and the biologists are very skilled at doing this. They isolate mast cells, which are the type of cell in the intestine of the pig that make heparin. And they actually characterized the full pathway and described all the enzymes involved in the biosynthesis of heparin. And so this is a complex product, but we have all the tools. These enzymes are like your wrenches. And we have all the tools, and all that we have to do, ideally, is use these tools to reassemble this outside of this intestine of the animal. So we looked at 3 approaches, and we're currently targeting 3 approaches. And this is the long-term approach, and that would be an approach that maybe Cynthia, my colleague, would take, and that would be to use a microbial community or use metabolic engineering. And this approach I'm taking with Matthias Kofus in my Constellation, who's a metabolic engineer, and that would be to take bacterial cell, either one or multiple bacterial cells, and engineer them to make this product in culture. And you could imagine large-scale fermentations producing 100 metric tons of this product each year. Unfortunately, you can see there's only one green arrow, one thing we've accomplished in a decade. 2 yellow arrows where we have made some progress, and 1 red arrow where we don't have a clue of how to make this work. And so this, I would say, is the way to go, but maybe the way to go in 10 years or maybe 20 years. So it's not an immediate solution to the crisis. How about a more near-term solution to this crisis? Here's an intermediate solution, and that's to use a biotechnological solution. This looks like, for those of you who are chemical engineers in this audience, it looks like how a chemical engineer or a biochemical engineer would think about this. They would think about using the enzymes as reagents and using vessels to do each step and running through all these steps and eventually isolating the product. And I'm working with this with John Dordick, and we have actually a center on campus. It's under the auspices of CBIS, the Biotech Center. It's called the Heparin Applied Research Center, where we have 12 employees who are actively working, moving this towards commercialization. So while the first long-term solution is still a research project, this is now a development project. So we can do it. We can make heparin using this biotechnological approach, but we can make 1 gram of heparin, not a metric ton. So we need to scale up 1 million or 100 million fold. And so this is a real development challenge. And we have to scale up at cost. We have to be cost competitive, at least close to the cost of the animal product in order to displace it from the market. So this would be our intermediate term solution, and we're hoping this is 5 years away to the market. The last approach is even closer to the marketplace, I believe. And this is actually not to build the whole complex drug product heparin, but just to build its active site, its active component. Just a few sugars, assembling a few sugars using these enzymes in a stepwise fashion. This is going to be a pretty expensive drug. It's only going to capture a very small, narrow share of the market, but it actually is something that we can do now on probably the kilogram scale. And it would be enough to go up to the 100 kilogram scale to capture that portion of the market. So this is pretty close because it's been tested and validated in vitro studies, this product, and it's even moved to specialized rodent studies. These are animals with kidney failure, so you even have to test in animals that have the same disability that the humans would have, that you would apply this drug, in the hospital too. And lastly, this has gone through primate studies, which are very expensive, very difficult to do, and the primate studies tell us this will absolutely work. And now this is at a point that we would call preclinical, and this is the point we would say we need an IND, and that would mean we'd go into patients with this drug, and we would do a clinical study and eventually go through a new drug application to approve the drug. So this kind of takes you through the stepwise approach to solving a problem using synthetic biology at RPI. Thank you.
So before we get to the next talk, it would be great if people who are standing to try to find a seat, it becomes easier, especially since we are videotaping this. So if any of you would like to sit down, that would be wonderful. Plenty of seats, especially up front. I know what happens in classrooms. Nobody wants to come up front. I promise I won't be nasty or anything. Neither will the next speakers. So the next talks, we're going to shift to the data science. given by two faculty. One, Jen Pazor. Jen is a professor in the Department of Industrial and Systems Engineering. And Jim Hendler. Jim is Tetherless World Professor of Computer Science, and he actually found an idea, or got an idea, to set up IDEA, which is the Rensselaer Institute for Data Exploration and Applications. Now, one of you needs this. Oh, no, you guys are fine. I'm not.
I'm Jim Hendler, and as John said, I'm the director of the Institute for Data Exploration Applications. And one of the things that Dr. Jackson has taught us, if you're going to have an acronym, be prepared to explain it. And I would be prepared to explain it for the clicker. Anyone know? So, the Institute for Data Explorations, clicker's not working, is, so we're doing something very different than a lot of our peers. So a lot of schools, because of the growth of data, and you heard that mentioned in the past couple talks, how data is really changing a lot of what we do, they're setting up a data science department or a data science operation. But what we've been doing at Rensselaer is a little different. We realize this is changing everything we do on campus, not just what we do in math or computer science, but all of our fields. So we set up this entity we call the Institute for Data Exploration Applications to represent the fact that the Institute represents that we're across all of the different fields. Our goal is to potentiate all of what we do here at Rensselaer. Data is the second word, and we're going to talk a lot about that over the next few minutes. But the World Economic Forum, on a panel that Dr. Jackson was actually on, came up with the phrase, the fourth industrial revolution. Said, we are going to see change over this next few years that's as big as the changes of the previous industrial revolutions being generated by data. Can you guys click to the next slide? I guess not. Exploration, right? Exploration is really the fact that it's not enough for us to just come up with ways of doing what we've been doing for many years with the data, just trying to analyze regressions and things like that. It's really that we need a whole new science. Hey. Right? So a whole new science called data science, and we really need to do basic research and to train all of our students across the entire campus in some of the principles of what it means to be doing data, and we'll talk a little bit about that as we go on. And finally, the applications we want to run really are linked to all these global challenges you've been hearing about, and I'll talk about that a little later. Well, okay. We got one. Can we get the next one? So I gave a TED talk a year ago. And I was supposed to be the second speaker. Instead, I was the first. And I came out, and this didn't work. So if you ever see my TED talk, you'll see at the beginning of my TED talk, I'm doing the same thing I'm doing today, which is ad-libbing while I desperately wait for my slides. Now we've got a primate study slide up on the screen. That's not going to help. So,
It's working on that.
Yeah. Well, except it actually showing Bob's slides.
No, that's not.
That's not, but the next one was. Anyway, there we go. Oops, had it for a second. No, so my slides are advancing here. You just can't see them, so ha. My next slide would tell you, you know, you're probably lucky you can't see my next slide, because it's one of those slides that's full of numbers. The way the previous talks have talked about orders of magnitude and things like that, my slide is an order of magnitudes of just how much data is out there now. We're at the point where each year the amount of data being generated is pretty much the same as all the data that's been done before. So over the next year, 2 years, we will have as much new data as there was data previously. And that includes big genomic databases. That includes manufacturing data. That includes just all sorts of things. In fact, this is a slide showing you a bunch of special issues of journals. Nature, the primary scientific journal in the world, has created something called Nature Scientific Data, an entire journal about this increasing kind of data we have. Next slide, please. So if you could see the slide, it would be talking about how this is really revolutionizing a lot of different things. In science, we're now referring to it as a fourth paradigm in science. So the first paradigm was sort of observation, right? You heard a great example of that in Karyn and Peter's talk, where they talked about the garnets, right? We're collecting things, we observe them, we can see what we can find. Second thing is we move to experimentation. So in our labs, we can actually generate the data we need, do comparisons, things like that, right? The third paradigm really started about the 1950s, and that was really moving to modeling and simulation. So the idea is we could use the computer power that was available instead of just, looking at an experimental set of data, we could try to model the system. We could get predictions. So we could say, if things are working this way, if this is how your heart beats, if this is how the heparin affects a system, then we're able to say, you know, now we can generate data and see whether it's doing the right thing. So that was computational modeling. So the fourth paradigm, and now we're back to our slides, making Jen happy, is data-intensive scientific discovery. So the idea is that by looking at data and correlating what we're finding and understanding the relationships it's showing us, that's a whole new way of looking at a lot of key scientific issues. Now, I've been talking about science, but let me just show you some headlines from recent papers and journals, right? How big data is transforming architecture, right? How big data is transforming social sciences, right? What big data is doing to business and what we do at the Lally School. In fact, we have now a master's of data analytics. And finally, it's making a big shift in engineering. And I'm going to hand off to Jen now and let her give you a good example of that with a working clicker.
So awesome. So supply chains are the complex networks that ensure the right materials, information, and money are exchanged to meet customer demands. And as our previous talk talked about the heparin crisis, when something goes wrong in a supply chain, that can be deadly. Other reasons why supply chains are important are, think about engineering marvels like a Boeing airplane. They require raw materials sourced globally to be manufactured in innovative processes and require thousands of technical people to be coordinated to make that airplane fly. And so supply chains are critical and important, but they're not a new concept. So supply chains have been around for decades, and industry and academic researchers have been thinking about optimizing supply chain decisions. However, today I want to talk to you about how supply chains are changing, and that change is due to the digitalization of how we live our lives and how the businesses operate. It's due to all this massive data that is available to us, and if we can really tap into that data to drive decision making, I believe the supply chains of tomorrow will be radically different than the ones today. And at Rensselaer, we're at the forefront of thinking differently about supply chain design. So across campus, across disciplines, we are thinking about data-driven supply chains. And if, there's a number of research projects going on, but I think two words really illustrate this research. The first is resiliency, which is the ability to withstand and withstand. Yes. Yes. Yeah. Thank you. The ability to withstand and bounce back from a disruption, which I think we're pretty resilient. And the current hurricanes in Puerto Rico, Texas, and Florida illustrate the importance and how critical supply chain resiliency research is. The first challenge is a logistical one. How do you get aid to the impacted areas? And at Rensselaer, we're taking a multidisciplinary approach to designing humanitarian aid research, and research here has led to policy implications that have improved recovery efforts. These are complicated problems because before we can offer the aid to the impacted areas, we need to remove debris from roads and we need to restore power. This means they're interdependent networks, and each of those different organizations are making their own decision making. And therefore, the power and the promise of data has shown with research at Rensselaer that if we can share and facilitate the right data, we can improve how fast we can get these networks up and running. The hurricanes have caused a second supply chain challenge. Did you know that 10% of all US prescribed medication is produced in Puerto Rico? And many of those are only produced there. Therefore, even if you are not in the impacted areas, you may be impacted by these hurricanes because of supply chains. And so research at Rensselaer into mitigation and preparedness is looking at how do you prevent this cascading failure where just a few key suppliers can have impacts all the way to the customer level. The second word about the research going on at Rensselaer is agility. Agility is the ability to be flexible and to change your decisions based on the current state of what's going on. So an agile supply chain is a data-driven supply chain that is able to take in real-time information and update operational decisions. So why do we need agile supply chains? Well, because today's customers are demanding it. You and I are expecting a wide variety of things to be delivered to your home. You are probably not willing to wait very long. You want them in not pallet sizes, but smaller. And, oh, by the way, based on recent surveys, not very many people are willing to pay much for that. Therefore, today's customer is fundamentally different than yesterday's customer, which aggregated at fixed store locations. Therefore, solutions that worked in the past are oftentimes too rigid for the customers of today. Consequently, today's supply chains are optimized for yesterday's customers. That means we really need to think differently about supply chain design. And it's really a disruptive time in supply chains. And I also think it's an exciting time. I teach the supply chain course, and I tell the world needs more data-driven, analytically thinking students to think differently about supply chains. And one way to rethink supply chains is to think about what we mean by capacity. So I ask you, do you think there's extra capacity on this road? Do you think we could move any more goods or people throughout this roadway? Any thoughts? Motorcycles? So if I ask that same question with this picture here, the answer is clearly yes. There's all kinds of underutilized resources available on that roadway. We can move many more humans or goods, but we need to think differently about what we mean by capacity. So we can't think about the car unit we need to get smaller. And we also cannot think about capacity as something that is just this fixed, owned resource. What if we can gain access to this resource? We need to operationalize capacity decisions instead of the typical tactical or strategic way they are today. And throughout the supply chain, there are many examples of underutilized resources. And this is really powerful. If we can use the data and the algorithms that are needed, we can tap into them and go from what is a very fixed supply chain network to a dynamic supply chain network. And research at Rensselaer has shown that this has improvements in efficiencies and resources because we're able to meet the dynamic needs of today by having elastic capacity, scaling it up and down where we need it. And we also have access to scale, which is increasingly important given today's customers wanting things fast. And so given we can get closer to our customers, that is a very powerful proposition. So I firmly believe the supply chains of tomorrow will be radically different than today. But this requires us to take all of that data that exists around us and truly use it to drive decision making. This requires us to think differently and think about new systems and processes about how supply chains operate, but it also requires fundamental research into data science and analytics and operations research such that we take this data and push it to drive decision making. And now I'm going to hand it back to Jim to talk about the challenges of doing so.
So when we talk about data, a lot of you, especially anyone who went to school before the past few years, are probably thinking about the stuff that lives in databases, Excel spreadsheets, things like that, things that are in nice, neat columns and things. But data today is much more complex. And so this science of data, of how we go from data to information, is becoming more and more of a driving problem. And part of the problem is the kind of scales we're talking about take us to a very different kind of data. So in looking at the problems you've heard about today, the microbiome modeling, the industrial production, Jen's supply chain examples, the problem is the data is not this nice, neat stuff where we know everything. It has a lot of different properties. One of them is that it's multi-scale. So a great example of that was when we were hearing about the biome problem, we were looking at things that were microscopic all the way up to the size of the Earth and eventually up to the universe, right? We're talking about trying to put, you can't just get every microbiome on the planet and put it in the right place and time and then model that whole thing. It just doesn't work. Never will. We have to find new ways of thinking about how we go between these different levels. If we're going to cure diseases, we have to figure out what's happening at the genomic level, but also how that translates into what you do at the behavior level, at the people level. The data is sparse. We're missing data. We don't know everything. We in a supply chain, and particularly in these kind of problems where we have these disruptions, how do you find the missing data? How do you guess what's in there? The old statistical models don't work for this new, complex, global scale data. It's distributed. It's coming from many different places. It may be different kinds of data. You're looking at images. You're looking at documents. You're looking at spreadsheets. But you're also looking at stuff that may come from a supplier who you know a lot about and a supplier who you don't know very much about. Something that's public data and something that's private data. They have very different qualities, provenance, et cetera. So we need to learn how to put together data from many different sources in new ways. And then a big problem is uncertainty, because we don't really know for sure that that data is exactly correct, right? We can't clean the data the way we can with data that we own and that we can check and where we know everything. But there's also a bigger problem. This is really the heart of this data science issue, which is that this data-driven science, this learning from the data, is correlative. It teaches us which things go together. Problem is, what we want to do in science is understand the causes, or in engineering, to understand the models. Say, if we change this, it will change that. So the bottom-up data and the top-down science look very different right now mathematically, and many of the kinds of models that Jen was talking about come about as we try to figure out how to reconcile these different kinds of math. So the approach we're taking at IDEA is to really say, instead of solving these as lots and lots of different problems, let's look at it as the things we do best here. So let's look at integrating data as a problem that all of our researchers can use. You'll hear another talk soon about looking at art and music and the mathematics of that, but perceptualization, how do we visualize, how do we hear, how do we interact with data in new ways? Data science and predictive analytics, I've talked a little bit about that, and I'll show you some examples in a minute, and also high performance computing. All of those things are leading us to a world where we can support these applications without each one having to do these things itself. So at Rensselaer, you heard so far about how we've been doing this in life sciences and healthcare, supply chain, environment, but we're also doing it for many, many other problems: cybersecurity, privacy, and resiliency of computer systems, data-driven physical systems that Peter talked about, understanding the built environment, architecture, new materials, new ways of doing things, materials and engineering, both manufacturing, advanced manufacturing, understanding what's going on in manufacturing processes, predicting new devices, social networks, and you'll hear later about machine learning and artificial intelligence. So without going into a lot of detail, these are just a few examples of some of the things we've been able to do where IDEA is helping campus researchers from many different fields to achieve their needs. So for example, in the healthcare arena, we've done analyses working with some of our local hospitals to help them make emergency room care better. We've worked with manufacturing processes, looking at both new kinds of manufacturing, such as the additive manufacturing that you hear about in 3D printing, and also how we know what's going on in big manufacturing machines where we can't open the machine to look inside. How do we take the sensor traces and predict what's going on? We looked at environmental data, and could we predict where invasive species may be going, instead of just looking at where they've been. And finally, working together with the Computational Center, the Center for Computational Innovations, or CCI, we're looking a lot now at a whole new thing. Some of you have heard terms like deep learning and machine learning. How do we take these huge, wonderful computers we have down at our computer center and use them for these data analytic problems? Requires new algorithms and new ways of doing things. So to summarize, the way we really view it is the way we view a lot of things at Rensselaer, that it's our people, it's our programs, it's our platforms, and it's our strategic partnerships with many companies that let us really do the research we do, or in this case, to really take and exploit the power of data. Thank you.
Thanks, Jim, Jen, and Jim. It's all my fault. I did that on purpose. I just want you to know that. So our next talk actually be the most complex of the set of talks, technologically complex, so hopefully this will work. So in this next one, we're going to be bringing together two very important things that people often don't think do go together, but they really do, and that is art and science. You know, we talk about Art X, the art in science and the science in art. You're going to see that here. Talk given by Jonas Brasch, professor in the School of Architecture, and Nina C. Young, professor of music here at Rensselaer.
Good afternoon, ladies and gentlemen. My name is Nina Young. I am a professor of music and multimedia composition and a practicing composer, and this is my colleague.
I'm Jonas Brasch from the School of Architecture.
And today we're going to talk about what art has to do with the new Polytechnic and the art experience, or Art X as it is coined here at the Rensselaer Polytechnic Institute. So the question is, how does art interact with the sciences and technology? Is this a new thing? Is it something we should be pursuing? Or is it something we've been doing for a long time? And I'd like to argue that this is part of a continuum. So we're going to look at these aspects, and we're going to talk about it through the lens of history, invention, innovation, and the human experience. And we're going to take you on this little journey of discovery through a series of time travels through space and time. So we're going to go back to very, very early human history, and then we're going to go and talk about the present day, and we're going to keep on going back and forth and following and playing against the arrow of time or processes of entropy, because music is a really special place where we can freeze time, go backwards, and go forwards.
And some of you might have wondered, when do we start to pursue art? And you might think that it came very late after agriculture, but the reality is art has really always been with us. So this is a map about human migration, and you see that around 50,000 years ago, the first humans came into Europe, and the oldest instrument that we know about is a flute that you see here down at the right, and it's a bone flute made from a vulture bone that's 40,000 years old. That's 4 times as old as the first evidence of agriculture that we have. And people built those instruments without any mathematical skills. So you might wonder, what does a flute like this sound like? How do they make the tone holes? And I think they made the tone holes just from practice. And we wonder, well, how could we sort of get an idea? We don't have, obviously, any music notation about the instruments, but we can just build one very simple from a PVC pipe. The material is not important, and we can play it. So we wanted to experience how it would sound like and imagine. And we have recordings from the impulse responses of the cave. So the people lived in the cave. That's the cave. It has 1.8 seconds of reverberation time. That's pretty close to a modern concert hall. And you can imagine that people played their flute in there. So we tried this. So it sounds a little bit different than a modern flute because the tone holes are very different. And probably making tone holes at the right position was one of the first mathematical skills that entered art. And Nina's going to talk a little bit about it.
Yeah, so how do we decide which pitches happen in music? Well, we don't really know historically. As a result of this flute, we can see that it was probably part of a trial and error experience. However, once we get to the time of Pythagoras, we know that people were systematizing using rules and mathematical principles to figure out how pitches would be in relation to one another. So anybody who studied physics understands the diagram on the left. Here we have a harmonic series, strings, and a node. So then we get intervals that you're all familiar with. We hear the octave, the fifth, the perfect fourth, the third, major and minor, and things like the seventh. And so here are perfect ratios. And we call this just intonation. However, if we listen to music that we think is very beautiful and familiar to us, such as this Bach example of the well-tempered clavier in E major, the prelude, we hear the word well-tempered. Well, well-tempered means averages and wrong. So if you look at the chart on the right, you'll see that we have these differences between perfect just systems, or mathematical ratios, and what we've done to approximate things. And this made different musical principles easy. However, if we go to the music of the present day, people are re-exploring these ideas of the past. So now we're going to listen to a small excerpt by a composer named Lamont Young, an American composer, a piece called The Well-Tuned Piano, playing on the idea of Bach. And here we have a well-tuned just intonation piano, also in the key of E, and it sounds radically different. So this brings up a discussion of what are we talking about when we see the differences between the past, the present, and using all of these different techniques. And why are we doing this sort of art at Rensselaer Polytechnic? Well, that's because the idea of a polytechnic involving the arts is not a new principle. If we go back to the time of Boethius, a 6th century philosopher, who is well known for really establishing the idea of the liberal arts. So you hear liberal arts and you're thinking about literature and paintings. Well, that's not really the case. The liberal arts are closer to the sciences that we know today. And there's a thing called the quadrivium. And the quadrivium is the study of numbers. And the elements of the quadrivium that you see below are arithmetic, geometry, astronomy, and music. Music being the study of numbers in time. So you can see that this is not a new idea. And in fact, music was used to describe planetary motion, systems, the cycle of the seasons, and even the systems of our human body. And music, as we just heard the two other examples, was an after effect.
So the question is, what does math and science have to do with music nowadays? We certainly don't want to get the harmony of the planet motion with musical intervals. That was old school. So what is Art X at the new Polytechnic? So Art X, the X stands for something. It could be science or engineering. And one of the things that we're really interested in is trying to get artistic processes into engineering thinking. So how could engineers and scientists and other disciplines benefit from art? So one of the things that might be different is that usually in engineering, things are goal-driven. I'm an engineer, I want to build a bridge, so I want to build a bridge one side of the river to the next side of the river. In art, often things are process-driven. You have a process, but you don't know the outcome of the process. We also want to combine different ways of thinking. In science and math, it's often very rational thinking. You try to think logically about things. In art, we often think intuitive about things. Can we combine both processes to make ourselves more successful? There's a big component in arts about immersion. Can we immerse ourselves in new environment that will inspire us? And that is going to be the next slide. So if we think about Gothic cathedrals, for example, we all know that Gothic cathedrals were built to worship God. You try to make them very high. But when you build a cathedral like this, the acoustics is also very, very different. It's very reverberant. You start singing in this cathedral, and the notes prolong through reverberation so long that the notes overlap, and you automatically get polyphonic music. That is something we discover through experience, and not something that we discover through rational thought.
So this is fascinating. If we listen to this music, we're hearing monophonic church chant. And you put this into the 12th century Gothic cathedral, and suddenly we get Renaissance polyphony. And polyphony is the independence of musical lines functioning together to make vertical simultaneities, and then we have harmony. This is one of the greatest inventions of Western music. So cultures around the world were using their experiences and their environments to innovate new ways to make art and music and to discover new things. And it happens that in the European continent, we did this thing called polyphony, which is really unique to us. But polyphony begs the question of how do we write these things down and how do we make sure that everybody is doing things at the times that we decide. So if we look at this inscription here, the Sekios epitaph, this is one of the oldest pieces of music notation that's available. And we see letters, so we know there's text and there's some melodic contours. But we have no idea how long everything's supposed to be and what the relations are in time. But then if we fast forward to the 14th, 15th, and 16th centuries, as you can see the manuscript on your right, we start to see things that resemble the modern music notation that we know today. And suddenly we're able to have multiple different streams of information all coordinating at once.
So we also sometimes think we directly map our perception to the world, and you are probably familiar with Escher's sketches. And this one, of course, is the illusion of the infinite ascending staircase or descending staircase. Things like that also happen with the auditory system, so you can produce a sensation of an always descending pitch. Mathematically, we know that's not possible, but I could play this for hours, and you always would think it would descend into basically nowhere because our perception is not directly mapped to the physical parameters.
So this is fascinating. So we can draw parallels to the common visual illusions that we know, and there are actually many auditory and oral illusions as well. And so now we are playing through the art experience with the idea of human cognition and perception. Now I'm going to talk a little bit about a personal artistic project that plays with these ideas. So we're going to return to this idea of time and the arrow of time and entropy. I was reading a book about sundials, and sundials are these pieces of antiquity, these timepieces that we used to tell what was happening. And the artisans of these sundials would always inscribe them with a memento mori, or a reminder that you will die. Rather morbid, but if we think about it, it's actually allowing us to realize that we are constantly in motion and moving towards something that we don't know what it is. So there's this quote that was found on an inscription on a sundial in Paris, and it translates to, "Life flies on like an arrow while it seems to stand still." Well, this is fascinating. How can something be in motion and stasis at the same time? Well, music is a place that we can do that. We can freeze time for you. We can go backwards and we can go forwards. And so I was looking for musical material, and I took the art of fugue, one of the most amazing pieces of the Western musical canon. And so here we have a fugue statement, so many different statements of the same idea, phased out in and out of time with one another. And I thought, okay, I want to use this as musical material, but this exists. How do I engage with the past? Well, I decided to freeze it. So I went into the studio, and I took that recording, and I time stretched it. And if you listen carefully, we're hearing the inside of the first note. And suddenly there are all these artifacts. And we can look into something that seems so simple and find lots of richness and detail. So after doing these different studio experiments and playing with the sound, I then mapped this to a string quartet like the original. And we hear a recontextualized version of that same music. And so here we're playing with a rather direct mapping of time. But what happens if we try to fast forward and think in advance of what somebody might do?
So we wanted to look a little bit into what we call interactive systems. So instead of having a score where you start at the top and end at the bottom, we wanted to develop systems that can react to other musicians. And one of the goals that we have, we wanted to build a device that can both do rational thinking and intuitive thinking. You know, if you remember, that's part of our ArtX idea about combining those two thought processes. So basically we built, with funding from the National Science Foundation, we built a system that has an auditory sensory system like a human does, all replicated in a computational model. It can then perform an auditory analysis analyzing two other musicians live. And then we have two processes. At one, we have a rational process that has an idea of music theory. You basically have a logic prover, and what you can do is you can start with a few axioms, and from those axioms it can derive its own rules. But we also have a bottom-up process that's more resembles intuitive thinking, working with systems like genetic algorithms, for example, where we can manipulate sound and time. So the system then can listen to two human performers and play with them as a trio member. And I want to show you a few examples. This is a trio we had with avant-garde music, free improvised, so we never played the same thing, with Doug Van Noort on the left, Pauline Oliveras in the center playing a digital accordion, and myself on the right. And this is the kind of music that we play. So the piano sound that's actually played by the accordion synthesizer. And this is another example just to get an idea about the variation.
This music sounds very different, but it's the same trio, right?
Well, it's the same session, it's just a different time part of it. And then what we did is we basically took Pauline out of the band, trained the music agent on her performance, and then used the material and altered it through the genetic algorithms so that it could listen to us too and perform with us. Now you hear the accordion coming and playing with us.
So it's amazing. You can play with Pauline even though she's not there.
Right, exactly. That's the idea. And this is another one. And here we have a dialogue between the saxophone and the virtual accordion. In the last example, we basically had Pauline play with her alter ego in the synthesizer. And if you have to watch a little bit her hand movements, so everything that she moves the hand, she's playing herself, and the rest is played by the agent, and at some point she stops and the agent finds a musical ending. So she stops. And here's your ending. And so the question is, what kind of technology could you apply this to? So one of the things we work with is communities with people with disabilities. So you can imagine if you have a mobility restriction and you have an agent like this, the agent can take some of your work. Actually, we have a symposium called the SATMA next week, and that's a 3-day symposium where we work with those technologies and enable people to perform music who would have a hard time otherwise. The other application that we work on is on jazz trainers. So I don't know how many of you have friends or have played jazz yourself, but usually what people do is they just use a so-called backing tape, and you always play the same recording of a jazz band, and you play on top of it. The problem with jazz is it's always different. And once you hit the stage, you realize that people play different things than on your tape. It's not like a classical orchestration. And so with an agent like this, the agent can play always something different, but always within the rules of jazz music, so the student can learn what is important and what is not important and remember those cues.
So Jonas, is there a way for us to play with space too if we can't be somewhere?
Well, that's one of the things, obviously you heard, everybody here heard about MPEG. That's one of the things that MPEG really was built for, is to really get this immersion. We talked about process-based experience, right, and research. And one of the things, for example, we had was the Gothic Cathedral. People probably would have never invented polyphonic music without having that experience. Actually, polyphonic music that's notated is something very unique about the Western world. So can we develop maybe even artificial environments where students and faculty and staff can just experience something and learn from it and come up with new ideas? And this is here one of the environments we simulate here with our trio, a system that has 45 seconds of reverberation. And in the last slide that we have, We also want to come back to MPEG. Remember when we talked about the concert hall with 1.8 seconds reverb, that was actually the cave. And the difference between a room like this and a cave is that the cave has non-regular walls, right? On a wall like this, I have a specular reflection, and on the cave, everything is irregular. So that's the one of the paradigms that we used for MPEG. So you see all those tiles. Those tiles really have the diffuse reflection like you find in a cave. We have to remember that our auditory system was probably developed 200 million years ago. When people inhabited that cave, they have the same hearing organ as we now have. That was never evolved on plain walls. So MPEG is really the space that takes the best of two worlds. On the one hand, we have all this modern technology, but on the other hand, it takes into account what human perception has really been designed for.
So we hope that this function is a little bit of a teaser and helps you expand your idea of how art can intersect with science and technology. And we would look forward to you coming and speaking with us afterwards, and we can tell you a little bit about more of our projects. And please sign up for any ArtX initiatives. It's really exciting material.
Okay, so we've heard a little bit about perception, and now we're going to hear some more in our final presentation today, given by Wei Su. Wei is a professor of practice in computer science and the director of our Cognitive Immersive Systems Laboratory. Also, he's a research scientist at IBM. And Selmer Bringsjord. Selmer is a professor of cognitive science and computer science, and also head of the Rensselaer Artificial Intelligence Reasoning Laboratory. What is it?
Perfect.
Yeah, I got it, right?
You want to give my talk?
Yes, I
better
will.
than I could
I
do.
will give your talk. now.
Okay.
So, who's getting this?
Oh,
No.
You are. Oh, does it work? Oh, yeah. It's
it should.
Okay. Okay, so I will be working with Professor Bringsjord to talk about the rise of Cyber-Sapiens. It works. So our human beings' capabilities and intelligence have been significantly augmented by technology and artificial intelligence. And this journey actually is going on. In a few years, we will be able to see beyond our visible domain. We will be able to see microwaves, infrared, with the help from hyperimaging technology and AI. So we can see what we cannot see. We will be able to use nanotechnology-based medical lab on a chip to monitor our bodily fluid in order to tell us if we need to go to see a doctor. We will be able to leverage machine learning algorithms and software for us to look at vast amount of data collected by billions of devices in order to understand our world better. And this journey goes on and on and on. We call it augmented intelligence. What's going to be the next challenge to us? How do we augment group intelligence? We always work together in groups. We create innovation, we create new knowledge, we transfer knowledge among us in groups. And we make decisions in group meetings, especially when we are dealing with vast amount of data. And we have to be able to immerse ourselves into the ocean of data and get more cognitive computing and artificial intelligence to help us to make better decisions when we face big data. So this is the next challenge we have. And RPI is taking this challenge. We have this Cognitive and Immersive Systems Lab in which we're focusing on creating cognitive and immersive situations room in which we can, the technology can help us to deal with business discussion in the cognitive boardroom, in the cognitive and immersive classroom, help teaching, and in the cyber-enabled discovery and exploration room, the room can help us to find scientific discovery.
And also, the room can help intelligent analysts to deal with the crisis and so on and so forth. So this is what we want to work on. The cognitive situations room will be built on top of the technologies we created and we leveraged from both IBM and RPI. The lab is in the beautiful building, MPAC Center. The very first use case I would like to tell you about is a cognitive and immersive classroom for teaching Mandarin as a foreign language. Mandarin has been one of the 5 most difficult languages to learn in our world. Large vocabularies, large character set, and tonal language. In order to read the newspapers, you have to get 5,000 to 6,000 characters, not words. You have to know that in order to read the newspapers in mainland China. And in Taiwan, if you want to do it, probably you will need to get 10,000 characters, not words. And it's a tonal language. It's 5 tones we have to learn. And we don't want to confuse people when we want to sell something, but people understand we want to buy something because the buy and sell actually using the same syllable, different tones. So, and for a lot of students who learn Mandarin as a foreign language, they don't have an immersed environment for them to do that when they are young. They don't have parents who speak a native language in their family. We cannot send everybody to China when they are young to study Chinese. So there's a need for a cognitive and immersive environment for the student to go beyond what they learned in the classroom for just grammar, pronunciation, and vocabulary. They have to be able to practice the normal daily life tasks and start to talk with, for example, intelligent agent in an immersive environment. So we created this environment. In this environment, some of you may see the demonstration in the morning about this Mandarin project in MPAC in the studio too. This environment can help students to practice what they learned in Chinese by talking to the avatar from the student. And the intelligent agent can switch language between English and Chinese when necessary, and also understand the multimodal interaction as an alternative way to interact from the student to the avatar and the environment. And we started with a restaurant scenario for the students who just finished Unit 4 in Chinese Level 1 class. They just learn something about the word, and then they can practice the language over there. The room will also be able to assess the results from the student and tell the student what to do next. Lastly, the room will be able to tell a story about some of the food items from the cultural context so that the student will remember better about the language. Again, it is a group environment. A group of students can go over there and practice. The next use case that we have as an example is in the cyber-enabled exploration and discovery domain. One specific use case is in cancer image diagnostic procedure. Several experts in that environment can work with the room to classify images, tissue images in cancer diagnostic context. The room will be able to allow the several experts to talk to the system, to set the parameters and select different algorithms and methods to process the images. And the several experts can talk to them among themselves, and the room will be able to learn from them from the whole procedure in order to get better diagnostic results and better accuracy and faster. So as you can see, there are several more examples, but the fundamental idea is we are building the situations room with one common infrastructure. The fundamental research we are working on is to build a bridge between the group of people and the supercomputers, intelligent cognitive computing modules, and so on and so forth. There are many of them. So we call them Society of Cognitive Agents. The challenge we try to justify is how these people interact with all these intelligent computing units. So first, we start from the human-scale interaction space. There are three more research areas we are going to focus on. We are focusing on. One is we need to use computer vision, speech, and so on and so forth to understand what's happening in the room among the people. And then we bring those information to the mind of the room level to give the room a mind in order to let the room understand what's the beliefs, opinions, and biases among people in the room. And do they have an agreement and so on and so forth. And what are the related context information the room can bring to the people beyond what they are talking about. Once the room finds out something, the room will be able to communicate that back to the people through the most natural way, which is the natural storytelling approach that we are going to take. I'm going to talk to you a little bit about more details in each area. In the area 1, for the spatial intelligence, we have been working on sensor technology that can understand how many people are in the room, where they are. And in the lower left-hand side, actually, using computer vision technology, the room can understand who they are. And one of our colleagues was trying to play a game with the room and tell the room that I'm Kang, who is another student. And then the room say, don't lie to me, you are Rob. And the room understands multimodal interaction, gestures, and speech together to understand what people would like to do. And the far right is a new technology we created among several students. The technology can understand by collecting verbal input and nonverbal input about how many agreements have been achieved by the people in this room. So that's a brand new technology. And the other thing that I want to talk about from the immersive narrative generation technology perspective is we can deal with a large data set and create story on top of that. So this technology has been leveraged to deal with some questions like, tell me something about Roman Empire. The technology will be able to give you a journal and several pictures it found from the database, and also automatically generate a timeline visualization and a geographic visualization to tell you a sophisticated story about Roman Empire. So the last piece that I want to talk about is the mind of the room. In this area, we are working on Professor Bringstroyd and his student are working on building the theory of mind mental models for the room. And Professor Li Rongxia and one of his students is sitting here. They are working on how to leverage machine learning to do better collaborative decision making. So with that, I would like to hand over the talk to Professor Bringstroyd for him to tell you more about how he can leverage AI technology to augment our intelligence to far more in the future. Thank you.
Thank you, Huang.
Yep.
We'll switch. We have a wonderful working relationship. I think that's safe to say. I have utter and absolute trust in Hui. So you were in charge of my slides. You didn't make any changes, did you? This is not my first slide. I'm going to run with it anyway.
Yeah.
We make it to Homo sapiens sapiens, and then what happens after that goes from biological evolution to what I think both of us think of as cognitive evolution, if we're going to head toward this new species. We have precursors first before we get there, and then after that, something really magical has to happen that I think is my responsibility, and that's when the AI is smart enough to be a near equal partner for humans. They team up and the new species that we're both aiming for is born. So that's the idea, right? All right, there's Wei and there's me. He's on that side, I'm on that side. I said we had a great relationship. This is the game plan overall, and we don't care about that. The Air Force has chipped in. IBM doesn't have bottomless pockets, so we have to go to the government a little bit. So, okay, now my question is to you, how much AI do you think we need to get to this new species? I think it's going to have to be a lot. I remember when those things first came out, good old-fashioned calculators. This takes me way back. This can't be enough for a species. This was my calculus teacher in 12th grade, and he had the first one in the school, and we'd throw these things at Mr. Cleary, and we were just amazed. But I'm sorry, that isn't enough AI to produce a new species. So one might say, okay, well, what else might cut it? All the rage today, self-driving cars. What are we dealing with here? Is this enough? I expect, I think, with all of you in the near future that this kind of thing will be entirely possible. I can say to the AI, can you get me to Thais in time? And the AI says, not only can I get you there, but hop in because it's driving the car. Also pays attention to my sartorial deficiencies. Perhaps my wife would have been in communication with the AI, but here it is, November 22nd, 2017, the actual performance of Thais, by the way, coming up at the Met. I'm a bit of an opera buff. My wife doesn't know how much money I spend on opera, fortunately. And the AI says, yes, I can get you there. It'll be great. I get in the car, and it's wonderful. Problem, still really no big deal. I'm sorry. This is going to happen, but from the standpoint of what you and I are aiming at, no big deal. Well, what about AI that can play games? Well, I think in the news, we've seen a lot of this recently. So this is the most recent success story for AI, Go. So the best Go player on the planet is, hands down, not a human being now. All right, so the question is, what about this? This does follow a well-worn script that AI is really comfortable with. It's very well-oiled. This is Tinsley losing to Chinook. This is Kasparov losing to Deep Blue. And this just keeps going, right? Now, I'm afraid my answer was, oh, and let's not, you would have been very angry if I forgot that one.
Yes.
IBM's Watson beat the best Jeopardy player on the planet. I tried to explain in '98 that as great as these machines are, they're nothing compared to the power of the human mind. And so my answer to you is still a negative. This is not enough to merge with us to produce a new species. But it's important to realize I'm not just saying this. There's a mathematical landscape we have that should anchor the position that I'm advocating. There are three spaces in front of you. There is the mere calculative thought, then superhuman stuff at the very top, and then in the middle, really significant human cognition. If we look at where these games are, Watson, working backwards in time, Go, Deep Blue, Chinook, and I would put all of today's machine learning in here because all of these things are working at the same level. And by the way, this is just to show you that these are not metaphors. These are not labels I came up with. These are well-defined mathematical hierarchies. They even have really specific measures that, of course, we needn't bother to explicate. But the question is, where do these things go? Well, here's where they go. They're all beneath that line. They're all mere calculation. This is not enough to produce a new species. So I'm going to give you an outrageous question that I know would be so demanding that if any AI could answer that question, it would in fact be sufficient to create a new species if paired with us. Nope, this is an age-old question. Is backwards time travel possible? Forward time travel we know is a non-issue. Our satellites already do it. That's why we have to reset clocks on satellites. But the AI probably knows everything in the internet, all history of science, and would say, Selmer, I thought you knew something about logic. Kurt Gödel already proved and gave as a birthday present to Einstein that time travel backwards is possible. And this is true. The AI would be right. This is a really simple animation showing you what Gödel did. The star is the time traveler, and the time is going along the vertical axis. The traveler takes off, goes very gradually. The light cones give you that traveler's orientation forward. So the cones show the traveler is always looking into the future, so to speak, but ends up traveling backwards. The entire thing is made possible by the fact that the outer cylinder is rotating. This was Gödel's trick, and at the time it blew everybody away. Now, it's nice, but I would say to the AI, all right, thanks for the information, thanks for reminding me. But, you know, since high school, people have told me there are paradoxes to time travel. Time travel's for the movies, not for real life. Well, Here's one of those paradoxes. I try to give it to you very quickly. You have to have your literary thinking caps on. There is a wonderful author known as Arthur in 2056. So he's an incredible novelist, and he's venerated. He's long dead, but in 2058, Chris sets out as the critic to go back in time and interview this amazing author. So they send him to do this. There goes Chris. He's going back, and all the way back to 1958. He sits down with Arthur, and he says, wow, you're an amazing author. You're venerated for decades and centuries to come. Can I see what you're doing? He shows him some of his work, and Chris says, this is terrible. Turns out the author is conceited, and he's fifth rate. Everybody thinks he's just absolutely terrible. The trick is that Chris brought his portfolio with him from 2058, things he had not written yet. Chris gets called back for whatever reason. He has to go back. He leaves the portfolio there, and you can probably imagine what happens. Arthur, our author, just copies what Chris left. But he never wrote it. The problem, though, is this goes around and around and around in a loop and has produced a very intense paradox. Where the heck does the great literature come from? All Gödel showed us is that we can go around in loops. Well, there's nothing that creates the great literature in the first place, and this is the paradox. Now, this is what the AI has told us so far, and this is what you were alluding to in terms of the students. There are some students working on this. And now you know where your tax money is going, by the way, in terms of the Air Force research. The AI says, and this is not verbatim true, but this is really what we're hearing. Selmer, I can't do this, I can't handle it. I need a new calculus that handles general relativity. And so over the summer, one of those PhD students did in fact present a new calculus that does exactly what the doctor ordered in this regard in Budapest at the Rensselaer Institute of Mathematics. So given that, stay tuned. We'll see if we can sort this out. Thank you very much.
Thank you.
Thanks, everyone, for coming. This was the first time we've done this, and I'm sure it won't be the last. Thanks very much.